That's just a little sideline there. So we're going to now move from the world of bigger stuff to little stuff. And Arturo De Lozonne is going to speak about endosymbiosis, how parts of cells came about as a coalition of other cells, and now they're basically part of us. And so, anyway, I'm going to have to find your PowerPoint here.
Well, you do. I wanted to extend a couple of comments on Claire's talk.
Okay.
So, you may want to take the opportunity if you encounter somebody who actually believes in Earth model of the world, that they are more advanced than we are, because they would basically argue that all life in the planet is derived from the few species, the few kinds that appear or that were saved on the Ark, right? So that they would say that, for example, all felines are derived from one pair of some kind of cat thing, right? And then from that pair, then you derived the cats, the tigers, the lions, the jaguars, the jackals, not the jackals, the ocelots, mix my kinds there. And exactly right. So, and if you look at the genetic diversity of all those animals and their diversity in morphology, all derived from two organisms 6,000 years ago, they are actually proposing faster evolution. Much powerful and faster evolution than we biologists are proposing. So we should be on the same boat and saying, yes, evolution happens. You think it happens really, really, really fast. I think it takes its time, but we agree at least that things evolve.
And there's I know a guy that has a theory of continental gallop. Continents dashing across the surface of the earth. But what I would like to ask the creationist, young earthman, is, okay, so how did the blind salamanders that are in caves in Texas get here from Mount Ararat? Must have had a seeing eye or something. Okay.
All right, so where's the
black dot?
I'll just keep that. All right, excellent. So, and you know, it is amazing that, so I'm a biologist on the UT campus, and I am not a big biologist, as Steve was pointing out. I study cells. I'm a cell biologist. I study cells and structures and proteins within cells. That's my favorite topic. And, you know, of course, working with my colleagues for decades, you know, this issue of whether evolution is real and is not even a question. So it is really remarkable to be in this day and age, 100 years after the publication of Darwin's book, and still have to encounter people that don't seem to understand it. And you know, we have days recently exposed on TV, and of course I couldn't stop myself from watching it. Just like, you know, you go see a scary movie, or you get thrills, just to see what kind of arguments are being put forward. And basically, if you watch the debate with Bill Nye and Ken Ham was basically saying, yes, it's all science, is all good, and you can see, you know, how things are happening, but that doesn't tell you anything about the past because you weren't there, right? So you just need eyewitness accounts, and if you don't have that, you just don't know what happened in the past, and that's what it boils down to. Which is maybe true. So let's assume it is true. So if you don't have a witness account of how you came to this world, you know, who are all your descendants back to, let's say, Adam and Eve. Let's accept Adam and Eve. if you don't have witness accounts and written records, how do we know that we are all descended from Adam and Eve? Maybe not. Maybe there were other people out there that, or maybe we just poof into existence as well. So we could use that argument. Or, of course, now in crime scene investigation, right? So we have many crimes in which we have no witnesses. Nobody saw it. So therefore, we cannot solve it? No, because not true. Yes, we can solve things because there's physical evidence that was imparted or present at the time of the events in question that we can explore now and reach conclusions based on that evidence. So, I want to tell you a little story about some portions of the cell, some organelles, some little structures in cells that I find very fascinating. I haven't worked on them myself directly, I work on other little structures, but I wanted to tell you a little bit about it because it's, to me, a just gorgeous example of the crazy things that go on in evolution. So how many of you have heard of mitochondria and chloroplasts? All right, so we have our work cut out for us. So if we were just to explore the structures of cells, so we are all made of cells, all our tissues are made of individual units that can be grown independently in the tissue culture. We can take a little patch of skin, dissociate it into the individual cells and grow them as individual living organisms in a dish. We have animal cells, plant cells, fungi cells, all kinds of bacterial cells. But animal cells and plant cells have something in common, which is that they have a lot of DNA encompassed in a large structure called the nucleus. And they have many little other structures inside the cell. I would work on this one here, and the lysosome. That's my also have a major organelle, very important, called the mitochondria, indicated in there, which, as you may know, is the powerhouse of the cell, generates a lot of molecules that are used for energy inside cells. And those are also shared by plant cells. So plant cells have a lot of unique features, a cell wall that make them rigid. They also have mitochondria. And in addition to mitochondria, then they have little organelles called the chloroplasts that, as you know, are used for photosynthesis. And thanks to those little buggers, we have enough oxygen to breathe and live. So the thing that when people started really studying all of this, really with the advent of better microscopes and started really investigating the structure, the internal structures of cells, this is about 100 years ago, at the beginning of last century, people began to discover these little organelles. And they noticed that both the mitochondria of animal cells and the mitochondria and the chloroplasts of plant cells look somewhat similar in shape, in properties, in behaviors, in the way they stain. They look similar, to bacteria. So here's a picture, artist's rendition of mitochondria, which are little structures, and bacteria, and they are roughly about the same size, roughly about the same shape, and they have a variety of similar properties. So back in the day, people didn't even know about DNA, and a lot of biochemistry wasn't done. They were just looking for similarities, and began to wonder, I wonder if those things that we find in our cells, maybe they are actually bacteria living inside our own cells, which was deemed as crazy talk, right? I would have said that's crazy talk at the time. And this is one of the main folks at the time. Russian scientists in 1905 or thereabouts proposed that actually both mitochondria and chloroplasts represented bacteria that had invaded cells and were actually living inside our cells. And he little evolutionary tree. In eukaryotic cells, nucleated cells, and he was postulating a separate origin of life for bacteria. Now we would say saw this recently, this proposed these days, that there was one invasion of bacteria that's all present living organisms, and that there was a second round of invasion of bacteria that affected only the plant side of organisms. But at the time, he was just proposing this pure morphology with no other pieces of evidence. And as Claire was saying, well, we can come up with hypotheses. This is what we as scientists would call hunches or ideas, proposals. We just call them hypotheses. So he came up with this hypothesis: the bacteria are just living inside. A few years later, about the same time, it actually was a common talk among a lot of people of the time, of these organelles that maybe are symbionts. Maybe these are bacteria that are living inside our cells, and we are sharing resources, and it's a mutual beneficial relationship. This guy has a funny reputation because Wallen was a professor in the University of Colorado in Boulder in medical school. And apparently he was a really tough teacher. Apparently he wouldn't lecture. He was a professor of anatomy. So he would just bring cadavers and just start dissecting. And he would have his students around the table. And if you answer, he would just shoot questions. Okay, what is this? What is that? And what is this connected to? And if you miss a question, if you gave the wrong answer, you would get slammed on the chest. How about that? So he was a tough guy, but he was following on this idea on the side of, are there bacteria inside our cells? So he set up to try to purify the mitochondria. So they call him the mitochondrial man in the university. He published this paper, The Mitochondria Problem, where he described experiments where he was able to purify the bacteria, the mitochondria from cells, and put them in a nutrient-rich broth, and he got them to reproduce and grow. And he published the paper, and he got poo-pooed by everybody else. And they said, nah, you probably have a contamination. That's nonsense. Turns out they were right. Mitochondria cannot be taken out of cells and grown independently in the dish. And probably because of that, everybody just started forgetting about it and just got lost in the literature. So 40, 50 years go by, and then a very young scientist called Lynn Margulis. She was actually Lynn Sagan at the time, because she was married to Carl Sagan, famous Carl Sagan. Piece of trivia there. Second piece of trivia. They were neighbors with my wife's family in Champaign, Urbana. So they got to know their kids when they were kids. So anyways, Lynn Margulis published this seminal paper where she had gathered a lot of evidence of not just morphological, but a lot of biochemical evidence, and more importantly, DNA sequence evidence that had been gathered by a lot of people. And she proposed that indeed the mitochondria and the chloroplasts. She also proposed another structure that turned out not to be in the end, but she proposed that mitochondria and chloroplasts are the descendants of bacteria that invaded primitive cells long ago, about 1 billion years ago. And given the track record of this hypothesis and the response from everybody else, she did not get a very good response. In fact, she had to submit this paper like to 13 different journals before she got it published. But what was good about her is that she had the evidence behind her, and she began to gather more and more evidence and publish papers, which is the way you get things accepted in the field, in science. So her example is a prime example of how science is done by having an idea. And everybody can have an idea, and a lot of ideas are not going to work out. That's okay. But those that work because the evidence accumulates, and then another person gets convinced, yeah, I think you're right, let me do an experiment. And then that person convinces a third person, and eventually everybody in the field of biology accepted that indeed, and this is what we now have in textbooks. Every biologist understands the evidence and why we say with confidence that these two cell structures, the mitochondria and the chloroplasts, are descendant from ancient process of symbiosis of bacteria with a very primitive eukaryotic cell, which is what is illustrated here. And the details are not clearly understood, but the large sketch is very definite, supported by the evidence. So the idea is that there were some very primitive cells that became engaged in a very mutual beneficial relationship with a bacteria, a symbiosis. So they started sharing resources, living together, and they would benefit from that. And then they became a little bit more intimate. They started getting in touch with each other so they could exchange materials easier, and then they wanted to do it a little bit better. So eventually the bacteria entered what we would call the cytoplasm of that primitive cell. Still a bacterium, but just living inside. And then through time, it lost the ability to grow on its own, so it became completely dependent on the host cell, and now became a part of it, an integral part of it. They were no longer two separate organisms, they were one combined organism. The idea then was that a second round of symbiosis occurred again, this time with an organism that already had a mitochondria, and then became engaged in a symbiotic relationship with a photosynthetic bacteria, which we encounter in the world today. More and more intimate, became internalized, and then eventually lost the ability to grow on its own, and the two organisms became one, a plant cell, a very primitive plant cell. So this is what the evidence tells us, and to be honest, if I were to express this to somebody out of science, I would say, that would be justified to say, this is crazy talk. How can you have two organisms fusing together, blend into one? What kind of evidence do we have, right? By the way, if you want to ask me any questions as we go along the way, or if there's something I say that is not very clear, or if I say it in my Spanglish accent, just interrupt me, all right? So what kind of evidence do we have that mitochondria and chloroplasts are indeed the remnants of an ancient bacteria that invaded ancient cells? So there's many lines of evidence, but if you were to see a detective show today in CSI, what is the one most important and most convincing piece of evidence that will convince anybody in the world that somebody did a crime? DNA, right? So it's almost like magic these days, right? If you find DNA, you know the answer. Well, we've got the DNA. So it turns out that both mitochondria and chloroplasts have their own DNA. Their own DNA that is separate from the DNA, the large piece of DNA that is in the host cell. All right. So this is a diagram of a mitochondria. It has a complex structure with lots of little parts inside. But inside, in the very core of the mitochondria, there's a little piece of DNA. That DNA has the information for some important components that function in the mitochondria. It has what we call tRNAs, transfer RNAs, something that is called ribosomal RNAs. Those are put together in structures called ribosomes that are used to synthesize proteins inside the mitochondria, just as if it was an independent living organism. Most importantly, the genes, the DNA sequence that you have in the genome of the mitochondria, the same thing is true for chloroplasts. They resemble, if you compare them with other organisms, they resemble those of the bacteria from where we assume they were derived. All right. So the mitochondrial genes are more similar to those of a specific type of bacteria, not any old bacteria, but a specific type of bacteria from where they are descended. The chloroplasts have a different sequence that is most similar to the photosynthetic bacteria from which they are derived. So we have the evidence that the DNA is telling us this is the leftover DNA of an ancient bacteria that is still present within us today. Isn't that amazing? It's just to think about that in our cells, in every single one of our cells, we have pieces of DNA from a bacteria that invaded our ancestors 1 billion years ago. Just gives you the chills. But why did it become dependent on the host? Why do we say that the two came together, that they fused together to form a new organism? And what happened is that the genes of the bacteria, many of those genes were lost, but many of the genes from the bacteria that became a mitochondria were actually transferred to the nucleus of the host cell. So our DNA, our host DNA, if we throw away our mitochondrial DNA, our nuclear DNA in it has actually copies of that bacterial DNA that were transferred from the bacteria to our DNA. So that's the diagram here. So here we have a cell, and we have the organelles, either mitochondria or a chloroplast. Yeah?
So we are genetically modified.
We are GMOs. We are GMOs. Don't eat each other, though. And the genomic DNA in the nucleus of the host cell contains all of those genes that were once in the bacteria but have now since been transported to the nucleus. There's still some organelle DNA that makes its own RNA to make its own protein synthesis. But the vast majority of proteins that function in the organelle, mitochondria and chloroplast, are actually encoded in the nucleus. They have to be made by this host cell, and then they have to be transported back into the mitochondria to put them there to do the job that they need to do. All right? So it's kind of like complex machinery now. It's not a simple thing, right? But it works, and it works beautifully. And we can trace where those genes were inserted, and see them right there. Now, that happened, as I told you, let's say 1 billion. Let's round it up, 1 billion years ago, both for mitochondria and chloroplasts. So obviously it was a very lucky case, just random occurrence of these two organisms fusing, and end of story. It would never ever happen again. Before I go there, Ken Ham would have asked me, well, how do you know that the genes were transferred? There's no mechanism to understand how genes would hop from one organism to the other. You weren't there. Maybe they were created in there already, right? So we as scientists we would say, well, you're right. I hadn't thought about that. Well, maybe let me try it. Let's do an experiment and see if it is true that genes can hop from one organelle to the other. So these are just two papers, recent papers from last year. There's a whole slew collection of papers that have people that have studied the process of genes hopping from the mitochondria and the chloroplast to the host DNA. And they're studying actually the molecular mechanisms of the proteins that do it, how it happens, and why it happens. All right, so we don't need to understand what they say. I haven't read them carefully, but there's a large literature with experiments today that demonstrate that indeed genes can hop from the mitochondria to the nucleus, and genes can hop from the chloroplast to the nucleus of their host cell. So we have a mechanism, and we have the effects, and we see the results of what happened of that hopping of genes 1 billion years ago. Now, of course, as I was saying, that was 1 billion years ago. Maybe it wouldn't happen again. So, and that would be true if you don't explore the world. If you just look at the textbook and see that in the textbook, and you say, well, that's crazy talk in the textbook that happened 1 billion years ago, end of story. Well, go out and walk into nature and start looking at organisms around you and see what you find, and you'll find that this is a continuing process. So what I'm going to show you is not in any textbook. So there are, for example, organisms that today they have mitochondria, they don't have chloroplasts, but they are right now engaged in a symbiotic relationship with photosynthetic bacteria. So this is a diagram of such organism. It's an organism called Polynella chromatophora, and it has its mitochondria and it has its nucleus, and it partners with photosynthetic bacteria, cyanobacteria. And we actually are beginning to see the transfer of genes from those cyanobacteria to the host nuclear DNA. This is the process of forming chloroplasts in our lifetime, right now, right here. All right, so what happened 1 billion years ago is still happening in this world. All right, so and we can see exactly what we predicted would happen 1 billion years ago. We're seeing it happening in the lab after collecting samples from nature. All right, so endosymbiosis is not a rare event. It's actually prevalent in nature. So prevalent that, in fact, once you start digging and looking in ponds and going around taking samples and looking at organisms, you find that, in fact, this happened not once, not twice, but maybe 3 and 4 times in a row. So now there are clear descriptions of what we call primary endosymbiosis, where one organism internalizes another one, like in the case of the mitochondria, right, or the chloroplast. Secondary endosymbiosis, where now we have another organism, a second organism ingesting or internalizing, say for example, a green alga that already has a mitochondria and a chloroplast, and now this organism becomes a part of the other one in what we call secondary endosymbiosis. And then there are examples of tertiary endosymbiosis, where this organism now would become an internal part of another larger organism. All right. And in some of those cases, they are sharing genes back and forth. All right. So it's a crazy world out there. All right.
So algae.
Yes, right. So the algae are just remarkable, and they are plentiful, and they don't mind sharing, So they are, since I don't know, we're going to have to talk about sex. These guys are doing, cellular sex across species. So what this reminds me, culturally, is basically of things nested in other things, nested in other things. Yeah?
The nested symbiosis, are these all prokaryotic or are these existing eukaryotic?
These are existing eukaryotic cells.
And are they ingesting other eukaryotic cells?
Yep. They are adopting other eukaryotic cells that have ingested other eukaryotic cells. Multiple rounds. So when you start doing a study of, if you cut a cross-section through the cell, and then you see a chloroplast, but then there's another membrane around it and another membrane around it with a nucleus in it. So these are a nightmare to study.
Already quite complex cells themselves depending on.
Exactly. Yeah.
So who makes the transmembrane proteins? Does the host nucleus or does the chloroplast make those transmembrane proteins for which?
In the nested ones, I don't think it's been studied biochemically that carefully. So a very important question. And how do you shuttle proteins back and forth through these multiple rounds of membranes? It'll be decades of work to figure that out. Yeah.
What are the projected implications as far as medicine goes and improving our health?
Well, in terms of health issues, I'm not certain. Now, a lot of these guys turn out to be parasites. So they definitely will affect our health. And now, here's an important news that I do remember, is that knowing that some of these organisms, which are parasites, have an organelle derived from a chloroplast, means we can use toxic substances that affect chloroplasts that we don't have, and therefore a chance of having secondary effects on our cells is diminished, right? We can attack them because they have chloroplasts, we don't. So that's kind of been a bonus information. And then, beyond that is just in knowing what's out there. Now, this is all in little bugs, right? So a bug is a bug is a bug. Algae, great. This would never happen, of course, in us, in larger animals, right? In larger things out there in the world. But of course, I'm here telling you this because I'm going to disabuse you of that notion. So it turns out that there is endosymbiosis in animals. Now you all know, I'm sure, and if you don't, you gotta know about this, that we have tons of bacteria living on us, in us, everywhere. Every orifice, everywhere, you have tons of bacteria. And the study of bacteria and organisms living in us is a huge medical area of research right now, because it affects everything from obesity to even psychological issues of not your genes, but the genes of the bacteria that are on you. So in our guts we have a jungle of microorganisms, right? But they are separate. We have our intestinal cells, which are ours, and then we have all the other bugs living in our intestine. But that's not true of many insects. Insects like aphids or termites, they have a lot of bacteria that help them digest. So here's a picture of aphids. An aphid has a unique collection of cells that capture bacteria and put them inside their cytoplasm and collect those bacteria, because those bacteria are useful for them to digest the plant material that they need to digest. I'm not a plant biologist, so I don't know exactly what they eat, but they're using for digestion, just like we do. Mealybugs is another nice example, where again in their cells they carry bacteria, not on the surface of the cells, but on the interior of their cells. And these very recent published studies found the remarkable discovery that in millibugs, they have not one, but two internal bacteria. All right, and here's in a very pretty picture, but let me show you a diagram of what this looks like. Again, it's another case of nested endosymbiosis. We have the intestinal cells of, here it would be a large intestinal cell of the millibug. Inside, they have large bacteria. The large bacteria is called Tremblia princeps. And that bacteria inside has multiple copies of a second type of bacteria. So the large yellow bacteria here is shown in blue. And then the other bacteria, the little bacteria is shown here in red. And they are inside the other cells. All right? So again, it's another case of Russian nested dolls. There's the one bacteria called Moranella endovia inside of Tremblia princeps inside of the millibug cell. So this clearly shows that endosymbiosis can occur not just in single-cell organisms, but even in multicellular complex organisms like insects. Could it be that it happens in more animals? Maybe we just need to start digging around and see what we discover. Right? So let me conclude just by pointing out the name, Moranella endovia. Moranella means little moran. And Moran is the last name of a scientist that discovered some of these organisms, and an active researcher who is doing this kind of stuff, who we just happened to recruit to the UT campus. Nancy Moran is an amazing scientist who does a lot of informatics and does this kind of studies. So if you have another meeting in the future, I highly recommend calling on her and have her come over and give you a talk. What they found, which is most remarkable, is not, it's already remarkable that there's cells within cells within cells, but what is published in this recent paper is that they have discovered that there is already transfer of genes occurring between, not only between the two bacteria, but they have been able to transfer the two bacteria, have transferred genes to the millibug DNA. So the millibug DNA already contains bacterial DNA. And the expression of those proteins, all proteins across, are necessary to carry out some specific biochemical pathways that are necessary for the three organisms to live. So in this complicated arrow diagram, you have the formation of specific proteins in each of the three organisms. And those are necessary for the survival of the three organisms. So they essentially, the three organisms are already one because they are interdependent on each other. So when we are asked, were you there? You can say, I am here. This is happening now. All right? And I'm going to go through it.
Questions?
Going back to the initial gene transfer from the internalized microorganism and its own DNA gets transferred or internalized within the larger cell's nucleus. The fact that there is some of the DNA gets left behind, is there any particular selective pressure that's deciding which genes become part of our nucleus and which genes get left behind in the mitochondria? Is there any pattern there, or is it?
Yeah, well, so that's an interesting question, right? So, which genes are transferred? Why haven't we transferred just all genes?
Yeah.
So it's been 1 billion years, but it's just transfer everything. So the answer seems to be that those genes that are left behind are really difficult to have imported back into the mitochondria for function. There's no mechanism evolved yet for those proteins to come back. So with that difficulty, you have strong selective pressure to keep them inside. When they're transferred, what is known is that the genes are not cut and pasted. It's a copy of the gene that is inserted first, and once that is well established in the nucleus, then you have freedom to lose it from the microorganism DNA. So there will be 2 copies before. And the thing is that if you look at different, this loss is going to be, of course, happening through time in different cell and organism lineages. So if you look, for example, at plants, at different plants in the tree of all plants, you can actually trace the loss of genes from their chloroplasts in different branches, but not in others.
Right, okay.
Do the genes that get moved end up with introns?
Sometimes they do, sometimes they don't. So a common mechanism for transport is by what we call retrotransposition. And that would mean most of those would be without introns. There can be secondary insertion of introns, that's been documented. And yeah.
Okay.
Question.
Oh, another question. Yeah, go ahead.
We have like bits and pieces of viral DNA in our DNA.
Lots.
Is there any known case of any of the viral DNA being used?
Yes. Many examples. So what he's referring to is that through our life history as organisms, we are constantly being infected by viruses. In few occasions, that virus makes it into our germline, so it's then passed from parent to child, and that's permanently integrated into our genome. Right? And that means all cells in the body contain the same copy of viral DNA. So I forget the proportion, but a third to half of our DNA is derived from ancient viral infections. So that ancient virus can be mutated, and there are few instances in which the viral DNA then adopts a new use. Prime example is in placenta, in the formation of the placenta, the early placental animals. There was the adoption of a viral protein that started having a second function. In many other cases, viral sequences start to turn on and on enough endogenous genes in different ways for novel uses.
So that'll be on the quiz.
Yes.
Thank you again.
You're welcome.